I'm an RU intern here at the beautiful Chicago Botanic Garden, and today I'll tell you a little bit about my project. With the help of my mentor Nora, we conducted the genetic rescue of Cirsium hillii, a thistle native to Illinois' gravel hill prairies. Genetic rescue is the increase in population fitness owing to the immigration of new alleles and can be an important tool for conserving rare plant species. Cirsium hillii is especially at risk of extinction because of its self-incompatible reproductive system, which means it cannot pollinate itself and must be pollinated by a genetically distinct neighbor. That means that when populations become smaller, the probability of a neighboring plant being inbred or a clone increases, reducing offspring fitness. When Cirsium populations are large, there is a lot of genetic diversity, little inbreeding, and a successful growth rate. We call the positive correlation between population size and fitness the allele effect. So the idea behind genetic rescue is that we transfer the alleles of the large, healthy populations and augment the genetic diversity of the smaller, struggling populations to boost their fitness. With higher genetic diversity, these fragmented populations will be better equipped at adapting to a changing environment. In practice, genetic rescue looks a little something like this. Every day, Nora and I drove 1 to 2 hours to our field sites and hiked through the prairies to reach our plant populations. We collected pollen from the Cirsium flowers using pipe cleaners and then used the pollen to pollinate half the flower of a distant plant, separating the pollinated side and non-pollinated side with a wire to keep one half open as a control. Hand pollination was done using a bee for better pollen transfer. After a few days, we returned to the flowers and covered the flower heads with mesh bags to contain all the seeds. We collected the ripe flower heads 2 weeks after pollination and brought them back to the lab to weigh the seeds of the pollinated side and the non-pollinated side to determine seed set differences as a metric for fitness. The heavier the seeds are, the more energy and food the parent plant invested into the embryo. The results are yet to come, but that sums up my project. Thanks so much for watching.